Shall we start, ma'am? Knowledge is the life of the mind. It has a beginning, but no end. Wisdom is the right use of knowledge. To know is not to be wise, but to know how to use knowledge is to have wisdom. A very good evening to one and all connected here. On behalf of the Marian Star Center and PG and Research Department of Zoology, St. Mary's College, Autonomous, Thoothukudi, we are happy to welcome you all to the national webinar on Helminth Parasites. God speaks in the silence of the heart. Listening is the beginning of prayer. Let's begin this session by invoking God's blessings. Prayer song. Before moving on to the session, we have few instructions for the participants. Dear participants, the feedback link will be posted only at the end of the session. Kindly fill the feedback form with correct credentials. Now let's move on to the photo session. Dear participants, kindly turn on your video for the group photo.
Ma'am, I have taken, ma'am.
Thank you, ma'am. Thank you, dear participants. Now, may I request Dr. Jessie Dias, Assistant Professor of Zoology, to accord the warm words of welcome.
A wonderful and pleasant evening to all. It is a great pleasure for me to welcome and introduce our distinguished guest, Dr. Lamanda Lingdo, Assistant Professor, Department of Zoology, St. Anthony's College, Shillong. Sir was awarded with MSc degree in parasitology from Northeastern Hill University. In his doctoral research, he investigated phylogeny and phylogeography of Fasciola species prevalent in mammalian livestock. His area of interest include biodiversity studies, animal conservation, and parasitology. To his credit, he has a DBT-funded project on the plankton community of water bodies of East Hasis Hills, district of Meghalaya, and DST SERV project on multi-gene molecular characterization of limnate snail species associated with fasciolasis. He has published papers in the reputed national and international journals, participated and presented papers in the symposium and conferences. Dr. Lindo has held various positions and extended his services as member of the editorial board for the Journal of Science and Technology, published by the Research Cell, St. Anthony's College, Shillong. He has actively participated in the national and international workshops training programs, symposiums, seminars, and conferences. He has organized national workshop in molecular phylogenetics and its application funded by DBT SERB. Sir has been honored with various awards and achievements. He has been endowed with Young Scientist Award twice by the Zoological Society of India, Kolkata on 29 July 2016, by the Indian Society of Parasitology on 27 April 2017, and Praxis India Ambassador by the Embassy of Republic of Poland. He has produced and directed the International Communicating Science Award-winning movie, The Midun Slayer, which won the first prize in the International Parasite Film Festival Award. We are really fortunate to have such a renowned resource person today. On behalf of the Star College program, Department of Zoology, and who have connected here, I accord a warm and hearty welcome to you, sir. I extend my cheerful welcome to our principal, Reverend Dr. Sister Lucia Rose, our secretary, Reverend Sister Flora Mary, deputy principal, Reverend Dr. Sister Kulandai Teres. I express my hearty welcome to overall coordinator and member secretary of DBT Star College scheme, Reverend Dr. Sister Arokia Genesius Alphonse, head of the department, Dr. Helminth Pasanga, and all members of the faculty and my dear students. Once again, I welcome you all. Thank you.
Thank you, ma'am, for entangling us with your heartwarming words of welcome. I'm delighted to invite the honorable speaker of today's session, Dr. Dabanba Lingdorf, Assistant Professor, Department of Zoology, St. Anthony's College, Shillong, to enlighten us on Helminth parasites. Over to you, sir.
Thank you so much. At the very beginning, I would like to thank the principal of St. Mary's College, Sister Lucia Rose, then the secretary, Sister Flora Mary, the convener, Dr. Helminth Pasanga, and overall coordinator, Dr. Sister Alphonse, and then also the organizing secretaries, Dr. Gemma Diaz, as well as Dr. P. Subhavati. So my topic for today will be on Helminth parasites. Though if with Helminth parasites, it's a very broad field, because helminths, when we talk about helminths, am I audible in the first place? I didn't ask that.
Yes, sir, you're audible, sir.
Okay, thank you. So yeah, so talking about helminths, you know that helminths, they are composed of two phylum, namely phylum Nematoda as well as phylum Platyhelminthes. So it's a big group as such. And in this particular presentation that I'm going to be talking about as of now, I'll be talking about the Digenea, the class Digenea, with regards to the class Trematoda of Platyhelminthes. And under this trematodes, or under Trematoda class Trematoda, I'll also be talking about specific group of trematodes. They are known as food-borne trematodes, which means that we get these, they are zoonotic, which means the disease can be transferred between animals to human beings. And they are being transferred in such way through the food that we eat. So that's why my talk for today will be on food-borne trematodiasis. So I would like to share my screen now, and please let me know if there is any technical glitches, because this side of sometimes the internet might be a problem. I hope and pray that nothing, I mean, everything goes right. Okay, can you see my screen?
Yes, sir, we can see.
Okay. So I'll be talking about the okay. So I'll be talking about the name of the topic for today is Faciliasis and other foodborne trematodiasis. Now, if there's any question as such, please, you can stop me in between and ask for anything that comes up in your mind. So when we talk about fasciitis and other foodborne dermatodiasis, which means that I'll be specifying on mostly fasciitis and also give you a brief of what the other foodborne dermatodiasis are all about. So when we talk about foodborne dermatodiasis, they are a group of diseases that are caused by the parasites such as liver fluke. So we have the liver flukes, the genus of Clonorchis, Opisthorchis, Fasciola. Some of you, maybe the names might be new. And then the lung flukes, Paragonimus, and intestinal flukes, Fasciolapsis, then Echinostoma, etc. Now with intestinal flukes, there are around 36 genera. So in my talk today, I'll be talking about Clonorchis, the liver flukes, Clonorchis, Opisthorchis, Fasciola, Paragonimus, and with regards to intestinal fluke, I'll only take example of one, that is Fasciolapsis, which is very prevalent in India. So these foodborne dermatodiasis, they're also known as FBTs, which I will refer to them as FBTs. They are, around over 40 million people are infected, and they're also known as neglected tropical diseases. So these diseases are neglected in the sense that when it comes to parasites and all that, they are very much neglected. Bill Gates has been funding a lot of neglected tropical diseases, but sad to say that even the ones that I'm talking about, it doesn't come up in the list of Bill Gates. So besides being a neglected tropical, disease as such, then there is another category which they fall under where people don't want to, sponsor as such. So again, I'm talking and enlightening people about all these other neglected tropical diseases that are very important for us human beings. So the WHO, the foodborne disease burden epidemiology reference group, they classified out of the many FBTs, they classify Clonorchiasis, Opisthorchiasis, Fascioliasis, and Paragoniasis as the ones that are of main importance. So when we talk about FBTs, we'll go straight away into Clonorchis. Now, Clonorchis is the genus, and the species that we're talking about here is Sinensis. So it has some resemblance with China, the name Sinensis. So this parasite is very prevalent in Korea, China, Taiwan, northern Vietnam, Japan, and Asian Russia. So it's mostly towards the Asian countries. And people get this disease when they consume raw or undercooked freshwater fish containing the larva or the larvae. So the diagnosis of this parasite or of this disease will be mostly the parasite's eggs in the stool. And since it is a liver fluke, naturally it will infect the liver as well as the gallbladder and bile ducts. The symptoms, well, it will be indigestion, abdominal pain, diarrhea, or constipation. So the symptoms are very much any symptoms of other diseases or other liver flukes, which always affect digestion. So sometimes people miss out that it might be a Clonorchis sinensis. And the International Agency for Research on Cancer has classified this particular organism as carcinogen since 2009. And globally, around 35 million people are infected, and treatment will be with praziquantel and albendazole. And of course, there is treatment, of course, but then the best way is always to prevent oneself from being infected with such parasites. So when we take a look at the life cycle, this is a general life cycle of Clonorchis. So it starts off with here, number one, as you can see, the embryonated eggs are present in the feces. So they are the diagnostic stage. As you can see over here, it's been marked as diagnostic stage. Now within the within the embryonated eggs also, there is the embryo, which will develop when it comes out of the human body. So they are being consumed by the snail, which is the intermediate host. And inside the body of the snail, this embryonated egg will then give rise to the larva known as Miracidia. From the Miracidia, it will become the sporocyst. They'll keep on dividing. Then sporocyst will give rise to many radia larvae. And then finally, it'll give rise to cercari. So which means plenty of larval forms are being produced and in huge numbers as well. Parasites, they have this inherent property of being very plentiful and multitude. So once the cercaria is being formed inside the snail, it comes out of the snail body and swim in the water, searching for its next host. So when it sees any fish, then what happens is that it is, by way of flow of water current, and also as a source of food for the fish, it is being ingested by the fish. Now inside the fish, this free swimming cercaria will become metacercaria, which is the other stage. So the metacercaria will be seen in the flesh or skin of the fish. And human beings, if they eat such fish which is infected with metacercaria of Pleunorchis, then what happens is that we get infected in the sense that if we eat improperly cooked or uncooked or raw fish, because as you can see in Asian countries, there is this craze of eating raw fish, and drinking some alcohol content with it, in parts of Korea and Japan, all these places. So this is a very dangerous thing to do. So please keep in mind, if you do travel to those countries and if you happen to be presented with such a delicacy, think twice. So this is what happens inside the human body. They exist in the duodenum, and then the adults are present in the biliary duct of the definitive host. Now, when it comes to India, Pleunorchis in India, it was first discovered in the bile ducts of a Chinese carpenter in Calcutta in India in 1875. But this is not counted as an Indian, documentation or record, because this particular person is of Chinese origin, so of China or Chinese origin, and probably he must have come from there. But then the first case was reported in 1934 by Ken Bakchi of Pleunorchis from Assam. Though we see Assam, but back then Assam in 1934 was undivided, which means the state that I belong to and even parts of Nagaland was under Assam. So which part of Assam we don't know, but at that point of time it was Assam. And then in 1998, Pleunorchis was also seen from a 10-year-old child from Bihar, though he was detected in AIIMS, New Delhi, but the origin of the person is from Bihar. So a lot of these cases that you find in news, in publications, though they are from a particular hospital or a particular place, but it's very important to note from which place the person has come, because that will give us a very clear indication of the parasite as well as the disease. So next is Opisthochiasis. Now, this is very similar to Pleunorchis. So in fact, with morphology, with their disease pattern and everything as such, it is very, very much similar to Pleunorchis. So a lot of people tend to miss out on these two parasites as well. They are present in Thailand, Laos, Cambodia, Vietnam, Germany, and this is also seen in Europe. And you get this by consuming raw, undercooked freshwater fish like Pleunorchis. Diagnosis is the same: parasites, eggs, and stool. Then it also infects the liver, gallbladder, and bile duct. Symptoms are very similar to Pleunorchis. And of course, this is also considered as a carcinogen. And 10 million people are infected, and treatment will be praziquantel or albendazole. And if you take a look at the life cycle, it is also very much similar to that of Pleunorchis, where you have the embryonated eggs pass out from the feces, which are the diagnostic stage, and then inside the snail they become Miracidia, Sporosis, Radia, Cercaria. Cercaria comes out, and then it becomes a metacercaria in the flesh or muscle of the fish. And people who eats undercooked or improperly or raw fish, then they suffer from this disease. And of course, they exist in the duodenum, which means the metacercaria, the cyst exists in the duodenum, and the adult is found in the biliary or the bile ducts. And when it comes to Opisthochis, there was no report in India as such, but then in the year 2015, this is from the lab that I am in also. So one of my seniors did this work. This is Opisthochis novarca, which was found from pigs of Meghalaya, but this is also zoonotic novarca. So there are some human, there's some people who are infected with Opisthochis novarca as well. So this is about Opisthochis. So next we come to another interesting parasite known as Paragonimus. And over here we have put forward Paragonimus westermani. Now this is present mostly in China, Philippines, Japan, Vietnam, South Korea, Taiwan, India as well, and Thailand. Now, in Africa, it's known as Paragonymous africanus. In Mexico and Central and South America, they're known as Mexicanus. And in the United States, Calicotti. So how do people get this disease? By consuming infected crab or crawfish that is either raw or partially cooked, pickled or salted. So a lot of the people who consume such delicacies, they are infected. Symptoms would be fever, chest pain, and fatigue, dry cough with rusty colored blood sputum. So it mimics similar to tuberculosis, the symptoms. That's why sometimes it's misdiagnosed as well. And then diagnosis will be eggs in the sputum or sometimes in the stool. And they also have specific and sensitive antibody tests based on Paragonymous westermani antigens. And treatment will be praziquantel and triclabendazole. Now, take a look at the life cycle. We have the unembryinated eggs. The others are embryonated, but this one is unembryinated eggs, which is passed out from the feces. Then they become embryonated in the external environment. Then the Miracidia hatch outside. Remember, the other two, they hatch inside the snail, but Miracidia here, it's present outside in the outside environment. Then they are being ingested by the snail, and inside the snail, they become the sporocysts, radia, and cercaria. So the cercaria is always the free swimming. So it comes out, and the cercaria invades the crustacean and insists into metacercaria. Now, a lot of this disease is seen especially with people who consume crabs or certain crustaceans. And the parasite, they exist in the duodenum and become adults in the lungs. So this is the parasite which is infecting the lungs. The other two which I've been talking about, they infect the liver, but this one is the lung parasite. Now in India, a lot of work has been done by Sir Singh from Manipur. And in fact, it was from two sides. One is animal paragonimiasis, the other one is human paragonimiasis. So with animal paragonimiasis, it was discovered in 1859, whereas with human, it started in 1981. Though the actual case was in 1919, but this was of a foreign resident, which is of Chinese origin once again. So we can't say that it's an Indian record. So the Indian record started in 1981. Of course, the pioneer work was being done by Sir Singh from Manipur. And so if you take a look at the distribution of this disease in India, the ones in blue is for mammalian paragonimiasis, and the ones in red is for the human. So if you take a look at human paragonimiasis, it's mostly documented from Northeast India. This is related with the food habit, the culture, and many things that are there in this side of the country. Nevertheless, there is also some sporadic cases as seen in Maharashtra. So this is about the three liver flukes. The two liver flukes as well as the lung fluke. Now we come to the intestinal fluke. Now the intestinal fluke, they are of 39 or 36 species, but one representative I'll be talking about is the Phaseolopsis or Phaseolopsiasis, the disease. Now this is present mostly in South and Southern Asia, pigs as well as in humans. People get this disease by consuming raw or undercooked aquatic plants with encysted metacercaria. So diagnosis will be parasites eggs in the stool, and it inhabits it's present in the intestine. And the symptoms include abdominal pain, nausea, vomiting, fever, and diarrhea. The treatment will be with praziquantel. Now, if you take a look at Phaseolopsis, now it starts off with unembryonated eggs. This can be passed from either human beings who are infected or from pigs. Now they become embryonated in the water, become Miracidia. Then they are being ingested by snail. Now inside the snail, it becomes sporocysts, radia, and cercaria. Remember, cercaria is always free swimming, so it'll come out. Now the free swimming cercaria larva will insist as metacercaria in the vegetation. So here the thing is this, that if people likes to eat a lot of herbs and all those things during the food, so it is very much important that people take care from which place, or if they wash, or they should wash them properly before consuming. So once the human or pigs can consume this vegetation which is infected with metacercaria or Phaseolopsis, then they exist in the duodenum, and the adults seen in the small intestine. Now, with regards to animal fasciolopsis, it is seen predominantly in Northeast India, Bihar, UP, Tamil Nadu, and human fasciolopsis is also reported from Maharashtra, Uttar Pradesh, Bihar, West Bengal, Assam, Odisha, and Manipur. So these are the places that that the disease is prevalent in India. So next we come to fasciola, which is fascioliasis. Now, with regards to fascioliasis, let's take a look first at the life cycle. This is also a liver fluke. Now, this is with the liver fluke, the eggs that are passed out from human as well as cattle or other ruminants will be unembryonated. So they are also the diagnostic stages. And in the eggs, they become embryonated, and then the miracidia hatch, and then the miracidia then will penetrate the snail intermediate host, and inside the snail, they'll become the sporocysts, radia, cercaria. So a large number of cercaria is produced. This cercaria again insists in the vegetation, and when people consume such vegetation which present with the cysts, or animals who consume such vegetation with cysts, then they become infected. And the immature flukes, they exist in the duodenum. In fact, everything happens in the duodenum excision. And then they are present in the bile ducts as well as in the liver, and they cause the disease known as fascioliasis. So my talk for today will be most, concerning fascioliasis. So with regards to fascioliasis, this is also known as the liver rot disease. There are two agents which are important here, Fasciola hepatica and Fasciola gigantica. Now, according to Mascoma, Fasciola has a global distribution, with Fasciola gigantica occurring mainly in Africa and Asia, but Fasciola hepatica mostly in Europe, South America, Australia, New Zealand, and Africa. Now, you see that in Africa and Asia, both these parasites are present. So there is sympatric occurrence between the two. And this is a very deadly disease, so to say, because approximately around $3.2 billion is being spent, and around 600 million animals are being infected. Now, we see a lot of work being done with animals, but less with human beings, because it's very difficult for people to get the samples, so to say, if people, they misdiagnose such infection with other diseases, so they don't come forward to the hospitals and report themselves. So if you take a look at the distribution of fascioliasis, now this, in fact, the whole entire world is being infected with fascioliasis. This is with regards to human cases that has been reported. And if you take a look at the taxonomic classification, of course, phylum Platyhelminthes, class Trematoda, and then I think all of you know this, but just a recap of all this. And the genus is Fasciola, and the species, of course, Fasciola species. So if you see morphologically, they are quite distinct. We have Hepatica, which is broad leaf-like, and then Gigantica, which is much larger and narrower. And the shoulders here, if you take a look at Hepatica, the shoulders are very prominent, whereas in Gigantica, the shoulders are not that much prominent. They're less developed. And the branching pattern of intestines is simpler. In Fasciola gigantica, it's much more complex. And the eggs are comparatively smaller as compared to that of Fasciola gigantica. So everything in Gigantica is big. It's more complex, it's big, and all that. So that's the difference that we see mostly when it comes to morphology. So with regards to India, it was first discovered in 1953 by A.K. Varma, and he said that it was Fasciola indica. But in 1957, Sarwar, he pointed out that this Fasciola indica is nothing much but a synonym of Fasciola gigantica. So Fasciola gigantica is the predominant Indian fluke, liver fluke, which is seen around the country as well as in Northeast India. However, I would like to point out that Fasciola hepatica was also mentioned, but this is mostly morphological analysis. But we tend to view this with a little bit of skepticism, because the next slide will explain why. Now, a lot of data when we check for Fasciola hepatica in India, we see that a lot of work has been done only by people on morphology. And if you take a look at the slide that I had presented earlier, Gigantica is narrower, slender, longer, okay? But then when we take a look at their description, it has been written as Fasciola hepatica. So there is a wrong description and a wrong record which has been put forward. And because of this, sometimes when we go abroad and present papers, and if people know that we are especially from India, they do present us with a lot of questions, asking us about the intricate details of everything. So because of such publications, then it really hampers the research in our country. Then when we talk about morphology, the work that was being done mostly is seeing with body length, body width, the ratio and all that. But when it comes to molecular markers, nowadays people are going to molecular taxonomy and molecular characterization for proper identification. So with molecular techniques, they use mostly mitochondrial sequences as well as the other ribosomal DNA sequences, but also nowadays NGS has come with full genome sequencing and all that. It has happened. So in the case of Fasula, highest resolution for accurate genotyping of specimens comes from both nuclear as well as mitochondrial DNA. So with regards to the work that has been carried out, this is a glimpse of Northeast India. It's very interesting because you start off with the snow-capped mountains of the Himalayas. You come to the lesser mountains and the hills. Then you also reach the plains and even the floodplains of the river Brahmaputra. So talking about altitude, you have a vast gradient of altitude, right from floodplains to the highest, one of the highest mountain ranges in the world. So this is a very interesting place if one needs to study something related with altitude, difference in altitude, how does altitude help, but also with regards to climate change. Nowadays, this is a very important area for studying climate change. Then with regards to the work that has been carried out, these are the different locations and the different animals that that the parasites were collected. And if you see the molecular methods that we use, the markers we use is the ITS2 as well as the CO1. And these two, we with CO1 and ITS2, sequencing is being done. Then the sequence quality checking is being done, the phylogeny is being done, as well as phylogeography. Phylogeography can be done either by networking or as well as with other softwares known as Beast and all that, SDVA Beast. But we're not concentrating with that part as of now. So we'll talk about this when we see the other slides later. Now, when we see the morphology, we see that all the parasites, the morphology resembles Fasula gigantica. The only exceptions are these three. Now the word A here stands for Assam, C for cow. This is number one because I have some isolates that are repeated. So you have Assam buffalo and all that. So G is for goat, Y is for yak. Remember this yak, it looks like Fasula gigantica. Whereas Mithun, if you take a look at Mithun, if you see in the previous slide of the animals that have been documented, the Mithun species, they look exactly like Fasula hepatica. Then even the Sikkim yak, the isolate from Sikkim yak also looks like it's Fasula hepatica. Whereas the Arunachal Pradesh yak looks like gigantica. So we know the others, we know that more or less they'll be gigantica, but from this collection, we see that there are two types of animals or organisms that we have. So the SEM studies were being done, and on analysis of the SEM studies, they were not they were inconclusive with regards to morphology and the other things that are related with SEM. So we take a look at the phylogeny. Now this phylogeny is being created with ITS2 by Asian with Mr. Bias, the software Mr. Bias. And if we take a look at this particular tree that we have here, we have these two clades. The two clades are present over here, and one clade representing Fasula gigantica and the other clade representing Fasula hepatica. Now with Fasula gigantica, these are the asterisk marks. These are the ones, these are under our collections. Most of them are clubbed under Fasula gigantica, but there are certain group or certain number of our isolates that are under Fasula hepatica clade. This will be Arunachal Pradesh yak. You remember that why Arunachal Pradesh? Remember Arunachal Pradesh, the morphology looks like Fasula gigantica. All right, so the morphology looks like Fasula gigantica, but when we look at the DNA, their DNA, especially with ITS2, we see that it is under Fasula hepatica. But with Sikkim yak, Arunachal Pradesh, Mithun, and the Mithuns and the yak from Sikkim and all, they are both morphology, representing Fasula hepatica as well as with the ITS2. So this is what we have with ITS2. But when we take a look at the CO1, the mitochondrial gene, now all of these isolates of ours are under Fasula gigantica. So which means that we have a mix, or an organism which is kind of mixed, especially the ones that we see with ITS2 that are clubbed under Fasula hepatica. So we have a mixed type of species. Of course, we have the ones that are purely Fasula gigantica. Then we also have the ones that show ITS2 of Fasula hepatica, but mitochondrial CO1 with Fasula gigantica. So on a perusal of literature, we found out that a lot of these also is happening across the world. For example, they have been reported in other parts of the world, such as in Thailand, in Japan, in Vietnam, in Iran. So around all these places where sympatric occurrence is there is the presence of another group of Fasula, which is neither Fasula hepatica nor Fasula gigantica. They have named this group of Fasula as Fasula species. So some of them, they've also termed them as hybrid or introgress and all that. But as of now, we've given them the name as Fasula sp. Fasula species, where they represent both from gigantica as well as hepatica in their DNA component. And when we take a look at the distribution, we found out that the haplotypes, when we do the median network analysis generated for ITS2 and MTCO1, we found out that a lot of that corroborates to what we see in the phylogenetic tree, which means that these median networks, they strengthen the phylogeny that we have created, because a lot of them, the cluster 1 will be of Fasula gigantica, whereas this cluster 2 will be mostly of Fasula hepatica. But if you take a look at the MTCO1, they represent major clusters with small branches here and there. And if you take a look at the geographical distribution of ITS2 and mitochondrial CO1, we know that Arunachal Pradesh is very much diverse when it comes to distribution of Fasula in Northeast India with regards to ITS2. And when it comes with regards to MTSO1, we know that Manipur is much more diverse when it comes to these types of flukes. So in our work, we know that just going by morphology doesn't work these days, because when we recall the picture that I had shown earlier, the photograph of all the collections, we see that one which is gigantica, but is showing us Fasula hepatica in the ITS2. So both of them, they are mixed species. Though we see this representing both morphology in ITS2, but when we look at the CO1 of one of these flukes that look like hepatica, they are in fact the Fasula species. So nowadays there is another term, there is an in-between, there is a hybrid between Fasula hepatica and Fasula gigantica, and that organism is known as Fasula species. Will it ever become a separate species on its own? It's a question to be seen, or an answer to be seen maybe in the coming days, years, or decades, depending on funding from funding or sponsoring agencies and the work that is going to be conducted by various scientists and people all across the world. So with regards to fasuliasis, it is how do people get this disease? By consuming raw undercooked vegetation or even drinking water contaminated with Metha cercaria. The vegetation you see here is mostly the watercress, mentha. Mentha is the one that a lot of us, in fact, in India, we always eat mentha. And now dandelion, then water morning glory, water lily, all these plants, the cercaria likes to insist. So this infection is, it can be either permanent or seasonal, and it's mostly seen in a rural than urban areas. And with so what it causes, I mean, the diagnosis, of course, you see the parasites eggs in the stool. It is present in the liver and the bile duct. The symptoms will be fever, abdominal pain, enlarged liver, and the treatment will be with triclopyrdazole. Now, this disease in India, it has been reported in Arunachal Pradesh, Assam, Uttar Pradesh, Bihar, and Maharashtra. Now, with regards to Arunachal Pradesh, Assam, Uttar Pradesh, and Bihar, now a lot of these people, they have been reported mostly from Delhi, so to say, because that's where they go and get themselves treated. And then the people that go and get themselves treated in such hospitals, big hospitals, are also the ones that are higher in the social category, which means that they have some amount, they're much more stable in financial matters. So that's why they do this, and then they can get themselves checked. But the other people who are slightly below or who are less fortunate with regards to financial means, so the disease is not well documented at all, because I believe there's a high rate of infection with regards to fasciitis, especially in this side of the country, as well as other parts of the country as well. So this disease is not well documented in the country, though a high index of fasciitis is suspected. This was being said by one of the doctors as well as scientists, Ramachandran, in his publication in 2012. So we almost towards the end of the presentation. And with regards to FBTs, we have high prevalence with fasciitis, fasciolopsiasis, and paragoniasis. So fasciitis is a liver fluke, fasciolopsiasis is the intestinal fluke, and paragoniasis is the lung fluke. Now, of these three, the ones that really target children is fasciitis as well as fasciolopsiasis. And the ones which are of low prevalence is opisthorchiasis as well as clonorchiasis. So, okay, now this is the end of the presentation, so to say, but I would just like to point out what has been done in our group earlier also, which may be interesting to you all. So I belong to the lab which was being headed by Veena Tandon. She's a Padma Shri awardee, and she's taking charge of the Biotech Park, which is in Lucknow. So in the year, this was in 2016, yes, in 2016, there has been cases from Tamil Nadu as well as Kerala, where people have complained of ocular inflammation. On closer inspection, they found out that there was a migrant cercaria. So cercaria entering the children or the people over there when people go for swimming and all, and then they enter through the eyes, and then they get locked inside the eyes of the children or adults as such, and then they causes this ocular inflammation. So this was being reported. So I mean, this is Procerovum, which is again, people will not hear much about Procerovum, because again, from parasites like a hierarchy, you have the common or the popular parasites, then you have the not so popular parasites, then you have parasites which are very docile and all that, but at some point of time they would like to express themselves. So here also I would like to say that Procerovum also expresses itself in the case of ocular inflammation, which is seen in children in South India. So with this, I would like to summarize by saying that foodborne trematodes have been reported from more than 70 countries worldwide, and the data on actual prevalence and burden is scarce. So FVTs, we have seen they are of liver, lung, and intestinal flukes. And of the FVTs, 2 are carcinogenic, that is Opisthorchis and Clonorchis. And fasciitis is present in all the continents except in Antarctica, and treatment will be mostly praziquantel or albendazole for Clonorchis and Opisthorchis. And with regards to treatment of paragoniasis will be praziquantel or triclopyrdazole, and treatment of fasciitis will be triclopyrdazole again, and praziquantel for fasciolopsiasis. So with this, I would like to end my presentation with a beautiful word in our language. It's called Kublai. Thank you. But it has a deeper meaning. When we say Kublai, Blai for us means God. So not only we say thank you, but we also bestow God's wishing or God's blessing upon the doer. So with this, I will end my presentation. And if there is any questions, I would like to take from here.
Dear participants, now the session is open for discussion.
Okay, I see one question in the chat box. Can we
Can I read it, sir?
Sure.
So can we use these after genetic engineering since it has wide effects, wide range of animals?
Yeah, you can use. I don't understand the question properly. Can we use
These after genetic engineering since it has wide effects on wide range of animals.
Can we use these? What are these? You're talking about the markers, or you're talking about the molecular? I know this. But anyway, I think going by what I understand from your question, surely we can use these molecular markers with regards to identifying or characterization of animals, and we can also develop specific markers when it comes to human fasciitis. Because the thing is this, that with human fasciitis, of course, you can get the diagnosis from the stool, but at times also we miss out whether it's either gigantica or hepatica. So developing specific markers is the need of the hour, so to say, to correctly identify which species, so that the treatment can be done properly.
Dear students, kindly post your queries in the chat box. Sir, one more question is there.
Can fasciitis be transmitted from human to human?
Human to human.
Yeah, so it can be transmitted from human to human if proper hygiene is not being followed. So if people live in an unhygienic place where they pass stool here and there, and they tend to drink the water that is being contaminated with the stool, so those eggs will again reach to human beings. And of course, that does not cause fasciolopsis in the disease, fasciolopsiasis in the disease itself, because it will cause some kind of allergic reactions. But if the stool that is being infected with the eggs, then it goes through the snails and then get encysted in the vegetation. And by chance, people drink water which is contaminated with such vegetation, or food which is contaminated with such vegetation, then of course it can be transferred from human to human. It all depends upon hygiene. Are there any more queries? Because a lot of people, they have this misconception of parasites that people who are non-veg, they are infected. But when regards to Helminth parasites, a lot of people who are vegetarian also in the danger zone, because as you can see, a lot of the vegetation, the encysment or the metacercaria which they insist on the vegetation. So if people don't wash their vegetables properly, or they eat outside food a lot, or they even if they eat in restaurants and all that, then they might encounter the disease. Which reminds me of Leander Pace, suffering from parasitic disease, because he eats, I think, in a restaurant and all that, where they bring these salads and all, which was contaminated with parasite eggs. Any other questions, if you have?
So one more question is there in the chat box.
So yeah, I see the disease prevalence is almost in all parts of India and Australia. Ramakrishnan, control measures, please. So when it comes to control measures, uh, with regards to these FBTs, the only way that people can control if they know what food they're eating, and not, in fact, when it comes to Clonorchis or Opisthorchis, eating raw fish is not proper at all. And then when it comes to Paragonimus, which is very much documented in India, eating crabs or other crustaceans, shrimps, and all those things raw, it's not the ideal way. So even if it's fried, if you eat in a what do you say, a fast food joint, please make sure that it's being cooked properly. Now, this disease, interesting thing about paragonimiasis or paragonimus, which mimics tuberculosis, the symptoms, in Northeast India is very much prevalent. Why? Because in Northeast India, we have this famous chili, which is the Bujolokia or the Naga Dorset, which is one of the hottest chilies. So there is a misconception of people over here that when we mix anything with that hot chili, anything that is present inside the animal and all that will die. So they used to take crabs, they used to pickle it with the chili, or sometimes they used to make chutney with the chili, with the hot chili, and then they think that the parasite or any other organism must have died. But sad to say, it doesn't happen. So a lot of the infection from Northeast India with regards to Paragonimus is because of eating, these chutneys or pickles and all that have not been done properly. They just ferment them and then keep them in the sun and all that, but that is not the ideal way. So the main way, the main control measure is, of course, knowing what you eat and what you put inside your mouth. So, and eating at home is always the best option. If you have any more questions, I will be very happy to share the knowledge that I have.
I think there is no more queries, sir.
Okay, thank you so much. Thank you everybody for patiently listening to me. I hope you must have gathered some information or learned something new. So that's all from my side. Thank you.
Thank you, sir. Thank you for a wonderful presentation. Ma'am, Jessie ma'am, shall we wind up the session, ma'am?
Hello. Come in, miss.
Ma'am, shall we wind up the session?
Okay.
Okay, ma'am. Now let's move on to the formal vote of thanks. Gratitude makes sense of our past, brings peace for today, and creates a vision for tomorrow. I deem it an honor and privilege to propose a vote of thanks on this momentous occasion. First and foremost, I would like to thank the honorable speaker of today's session, Dr. Damanba Lindop, Assistant Professor, Department of Zoology, St. Anthony's College, Shillong, for his vibrant presence and wonderful presentation on Helminth parasites. Sir, the knowledge you shared regarding foodborne dermatodiosis is rare and informative. I hope all the participants have gained more insight into the topic. On behalf of the Marian Star Center and all those who have connected here, I convey my hearty thanks to you, sir. I also express my heartfelt thanks to our Secretary, Reverend Sister Flora Mary, Principal, Reverend Dr. Sir Luciaros, Deputy Principal, Reverend Dr. Sir Kolandi Teres, Overall Coordinator and Member Secretary, DPT Star College Scheme, Reverend Dr. Sir Aureki Genesius Alphonse. I also extend my sincere thanks to Dr. Hermin Pasanga, Head and Associate Professor of Zoology, Dr. Jessie Dias, Department Coordinator, and members of faculty for their constant support and encouragement. I accord my sincere gratitude to the technical team for their unstinted support. I would like to express my deep sense of gratitude to the enthusiastic students for their active participation. Once again, I thank you one and all for your cordial cooperation. Thank you. Dear participants, kindly note the feedback form is shared in the chat box. Fill the feedback form with correct credentials. Once again, I thank you one and all for joining us today this evening. Thank you.
Thank you, everybody. Good night. Take care.
Thank you so much.